Hi everyone, welcome to the poster session of ISMB 2020. My name is Doughan from KNUST Ghana, and I am presenting on the topic Unravelling the Transcriptome Landscape of African Burkitt Lymphoma Patients. My talk will take this outline. So before I begin, I would like to advise viewer discretion for the image on the next slide. So this is a picture of a child with Burkitt lymphoma. Burkitt lymphoma is a blood cancer that affects mostly children in Africa, and it always affects more males than females in a ratio of about 4:1. That's males to females. Now, BL is always linked with malaria and Epstein-Barr virus. Now, these risk factors are very prevalent in Africa. Therefore, Burkitt lymphoma is also very common in Africa, and it accounts for about 36% of all childhood cancers. There is inadequate genetic data on African individuals, and this impedes a targeted immunotherapy for Burkitt lymphoma treatment. As a result of this study sought to identify differentially expressed genes between African Burkitt lymphoma patients and non-BL individuals, and also to find the pathways these genes are involved in. Okay, so for the methods, the data used in this work was downloaded from NCBI SRA, and this was deposited by Abati et al. We checked the quality of the data using FastQC and MultiQC, and the reads were then aligned to the human reference transcriptome using the Callisto aligner. And for downstream analysis, we use 3 R packages for differential expression analysis, and then we moved ahead with the consensus genes from all the 3 tools. The final step was gene enrichment analysis, and here we used gprofiler and David annotation tools. Okay, so now concerning the results and discussion, I'll just run through a few of them. So after mapping the trim reads to the human reference transcriptome, we found that over 80% of the reads mapped to known genes. Then we moved ahead to perform differential expression analysis using HR, DSEC2, and Limavum. So DSEC was able to identify about 6,643 genes, while HR and Limavum also identified 2,639 and 2,370 genes respectively. So for the gene enrichment analysis, we used a consensus of genes from all the 3 tools. So we got about 1,765. So these are the genes we used in the final phase of our work. Alright, so this figure shows the output generated from gprofiler, and it gives a summary of the various gene ontology domains that the identified differentially expressed genes are involved in. So here we paid more attention to the KEGG and reactome pathways, where about 6% of the genes were involved in arthralgia and collagen formation pathways. Now, these pathways also included the gingival pathway involved in gum development and growth. All these pathways can be found around the jaw area of the body, where Burkitt lymphoma usually starts. Okay, so in conclusion, after analyzing RNA data from Burkitt lymphoma patients, we identified 1,765 genes, and we found that the most significant genes were the FECR2, CCF19, CO3A1, and these are all involved in gingival overgrowth, arthralgia, and collagen formation. Okay, so this is just a brief summary of my work. I would like to say a very big thank you to KUSC, where I am right now, Fugati, H3A Bionet, and Wasselbury. Many thanks for your support. I'll be online to answer all your questions, and I'll also take any ideas you have for me. Okay, thank you very much for your time.